Hello everyone. The question says a zoologist recovered some tissue from preserved skin of a woolly mammoth. Further genetic analysis requires DNA isolation and increasing its amount. Which one of the following techniques would be most useful for increasing the amount of DNA? The options are RFLP analysis, PCR, electroporation, and chromatography. As you can see, the question very clearly mentions that a technique that relies on the principle of increasing the amount of DNA. To answer this question, we can try to look at the definition of each of these options individually. The first is RFLP analysis, or restriction fragment length polymorphism analysis. In this method, we detect the differences in homologous DNA sequences by the presence of fragments of different lengths after digestion of those DNA samples with specific restriction endonucleases. So as you can imagine, we will have the extracted DNA that will be amplified by PCR, and then they will be digested with very specific restriction enzyme which will give rise to different length of fragments. In this, we also use an RFLP probe, which is actually a labeled DNA sequence that hybridizes with one or more of these fragments of the digested DNA sample, after which they are separated by gel electrophoresis, which will then reveal a unique blotting pattern which is characteristic to the specific genotype at a specific locus. So as you can see, this technique relies more on the downstream processing of the amplified DNA. The next option is PCR. PCR, or polymerase chain reaction, is a method which makes many copies of a specific DNA region in vitro. This method relies on the use of thermostable DNA polymerase and requires DNA primers which are designed specifically for the DNA region of interest. And as the name suggests, it's a chain reaction. So here the reaction which is carried out by the polymerase is repeatedly cycled through a series of temperature change, which allows many copies of the target region to be produced. Third option is electroporation, which is actually a physical method of introducing genetic material or proteins or drugs into the cells by the use of electric pulses. With the use of electric pulses, there will be these temporary pores created in the cell membrane which will enable the delivery of the molecules into the cell. So it is more of a delivery method than to do anything with DNA of the cell. The last is chromatography, which is also a separation method where the analyte is combined with a liquid phase or a gaseous mobile phase, which is then pumped through a stationary phase. And the components of the analyte interact differently with these two phases, and as a result, based on their polarity, they are separated out eventually with the passage of time. So you have all the components of the analyte separated from each other. So coming back to the question, the option that would be correct will be the one which increases the amount of DNA, and that would be PCR. To know more about PCR, you can refer to Basics of Biology course offered by NPTEL. Thank you.